Hi and good evening. So our group will be present about a genomic analysis provide novel insight into diversification and taxonomy of Alorhizobium vitis. So this is our group member, which is me, Emilia Natasha, Nurhana Humaira, Nurul Ayu, and Shasha Shamina. My name is Emilia Natasha and my matrix number is A195441. So I will talk about the background and the objective of this study. Alorhizobium vitis is a gram-negative, root-shaped bacterium with flagella that causes a crown gall disease of grapevines. This disease leads to economic losses in vineyards worldwide. Alorhizobium vitis was originally classified as Agrobacterium biva-3, but chemical and DNA studies show that it didn't quite fit in the Agrobacterium biva-3. Later, MLSA and genome-wide analysis confirmed it was genetically distinct. As a result, it's now placed in the genus Alorhizobium. Why is it matter? Understanding its taxonomy is crucial. It helps with managing crown gall disease, taking outbreaks, and protecting grapevine health. That's why deeper genomic and phenotypic analysis is so important to propose a new taxonomic classification. There are four objectives in this study, which is the first one is to investigate the genetic diversity within Alorhizobium vitis by sequencing and analyzing the genome of 11 additional strains. Next, to perform comparative genomic and phylogenetic analysis with an aim to understand the evolutionary history and divergence within the Alorhizobium vitis complex. The third one is to access phenotypic traits of representative strains to see whether genomic difference corresponds with phenotypic distinction. And lastly, to revise the taxonomy of Alorhizobium vitis by proposing the designing of a novel species within the complex named Alorhizobium apellinum.
Next, we move to the methodology used in this research. First, bacterial strains and DNA extraction, where genomic DNA was extracted from bacterial strains grown on King's medium B at 28 degrees Celsius for 24 hours using the nuclear spin microbial DNA kit. Next, genome sequencing and assembly. Draft whole genome sequences were obtained for 11 Alorhizobium vitis strains using Illumina MiSeq and NextSeq 500 platforms. De novo genome assemblies were performed using the Spades genome assembler. Third, we have genome annotation. Genome sequences were annotated using PROCA and the NCBI Prokaryotic Genomes Annotation Pipeline (PGAP). Fourth, phylogenomic analysis. Whole genome sequences of 69 Rhizobiaceae strains were used for core and pan-genome phylogenomic analysis. Next step is overall genome relatedness indices. Different auguries were computed to differentiate strains. For genome gene content analysis and clade-specific gene identification, pan-genome analyses were conducted on more focused datasets using two pipelines, which is Pentagrel and GAT homologous. Next is biochemical tests. Metabolism of 4-hydroxyphenylacetic acid and gentistic acid was tested in 80 minimal mediums supplemented with indicated. Lastly is MALDI-TOF mass spectrometry analysis, which MALDI-TOF MS was used to characterize and phenotypically distinguish the strains.
Hello everyone, so I will present the result of this study. So basically, as you can see from the figure above, 11 new Alorhizobium vitis strain was sequenced, and then the size is ranged from 5.67 to 6.52 MB with GC content of 57.5 to 57.6%. For the core genome phylogenetic analysis, 14 Allorezovium vitis strain revealed that it is not a single species but a complex species composed of at least 3 distinct genomic species. And then the species complex are Allorezovium vitis sensu stricto, which is clade A, and then the second is Allorezovium ampellinum species North, clade B. This is the new species, and then a separate lineage represented by strain AV2. Overall genome relateness in this ANI and DDDH confirm species boundaries among the major clade. For pan-genome analysis, it is identified 10,501 orthologous gene cluster among the Allorezovium vitis strain with 3,548 forming the strict core genome. And then for the clade-specific gene were discovered for each group show functional divergence and potential ecological specialization. Next, Allorezovium vitis sensu stricto have gene involved in the degradation of salicylic acid, genthistic acid, and 4-hydroxyphenyl acetate. Meanwhile, Allorezovium ampellinum contain gene related to polyamine metabolism, lysine biosynthesis, and nickel uptake. Furthermore, a significant number of species-specific genes in Allorezovium ampellinum were located on plasmid, which highlighting the role of plasmid in adaptation. Various types of tetrad utilization TAR gene cluster were identified across strain, but TAR region type was not specific to any species. Biochemical assays showed that Allorezovium vitis sensu stricto strain could metabolize 4-hydroxyphenyl acetate. Meanwhile, Allorezovium ampellinum could not metabolize it. And then Allorezovium tested strain including lacking identifiable TAR gene were capable of utilizing tetrad to produce alkali. MALDI TOF mass spectrometry analysis effectively distinguish between the genomic species supporting the genome-based taxonomy. Next, we move to the discussion slide. So the study confirmed that Allorezovium vitis is a species complex and it is also composed of a multiple distinct genomic species. A new species is Allorezovium ampellinum species North has been formally proposed based on phylogenetic evidence and genome similarity matrix. The existence of additional lineage, clade C, D, and E, suggests that more novel species within the Allorezovium vitis complex are likely to be discovered in future study. And then this pattern of species diversification is consistent with previous finding in Agrobacterium tumefaciens, which was also reclassified into multiple species using genomic data. And then finally, the clade species gene show that different species within the complex are ecologically specialized and potentially occupying distinct niches or responding to different environmental conditions. That's all from me. Thank you.
In conclusion, this research study made several significant discoveries about the Allorezovium vitis species complex. Firstly, this study had identified gene clusters specific to Allorezovium vitis, providing a deeper understanding of their ecological roles. These gene clusters help explain how these bacteria interact with their environment and host plants. Secondly, found that Allorezovium vitis possesses genes linked to stress tolerance and nutrient metabolism, highlighting their impressive ability to adapt to different conditions. Also demonstrated clear genomic differences between Allorezovium vitis sensu stricto and Allorezovium ampellinum, which not only confirms their classification but also reveals their unique adaptations. Furthermore, this study emphasized the importance of comparative genomics in understanding bacterial evolution and adaptation. By comparing genomes, we can uncover how different species adapt and evolve over time. Finally, these findings establish a strong foundation for future research in plant-associated bacteria. This can lead to better understanding of their ecological impact, potential benefits, and applications in agriculture.